Welcome everyone to the ARO seminar series centered around the topic of auditory nerve. Today we are delighted to have Dr. Mark Rutherford as our speaker. And my name is Juan Goutman. I'm from Buenos Aires, Argentina, and I will be your moderator for today's webinar. Before starting today, if you have any questions...
Yeah, that's the ARO talk.
Okay. If you have any questions during the presentation, please put them in the chat. After the talk, feel free to virtually raise your hand, to unmute, and directly ask your question. Any questions that are not addressed during this session will be answered by the speaker in a follow-up email. Closed captioning for this meeting is available. And this session is being recorded and will be made available for additional viewing. So I will provide some background about Dr. Mark Rutherford at the moment. Mark is graduated from the University of Missouri in Columbia in 1999 with a degree in nutritional sciences before heading to the University of Oregon in Eugene for graduate school, where he began to study synaptic transmission in the frog cycle. He graduated with his PhD in 2005 when he remained in the lab of Professor Bill Roberts for his first postdoc. In doing so, he switched from the presynaptic capacitance measurements of exocytosis to postsynaptic measurements of excitatory postsynaptic currents. In 2009, he left the US to do the second postdoc, now in the lab of Professor Tobias Moser at the University of Göttingen in Germany. He remained there for many years and was very productive in publishing many papers. And that's where he began working actually in the mammalian cochlea, where he remained in Göttingen until 2012, focusing on postsynaptic recordings and super resolution microscopy. In 2013, he founded his own lab, firstly as an instructor at the Washington University in St. Louis, Missouri, then promoted to assistant professor. His family is still in St. Louis at the moment. By 2023, he was promoted to associate professor with tenure in the otolaryngology department. Throughout his career, since grad school, Mark has remained focused on synaptic transmission in the inner ear with patch clamp, cellular electrophysiology, microscopy, and in vivo auditory neuroscience. Mark enjoys extensive collaborations with investigators in St. Louis, across the US, and around the globe with the team science approach. One overarching goal of the Rutherford team working in the WashU Center for Drug Discovery is to develop a drug for glutamate excitotoxicity in the inner ear and more generally in the CNS for mitigating neurodegenerative disorders. I just wanted to add that I'm a first-hand witness of this spirit of the Rutherford Lab in terms of collaborating across the world, as we've been collaborating now for almost 10 years, I would say, and probably been friend to Mark even more time. So Mark, without any further delay, please go ahead.
Thank you very much, Juan. I appreciate that introduction very much. Okay, so I think everybody should be able to see my screen now. So, right, I'm going to talk about synaptic transmission and spike generation in the cochlea. And this is a cartoon of my favorite synapse, the ribbon synapse, which I guess I've been studying in different species for over 20 years now. And for a brief outline, today I'm going to talk about these three things. And the first thing I want to say briefly is, and most importantly, is about the love of science. Science, you have to love your science, or else probably it's going to be hard to get anybody else to love it. And science is about studying the relationships between things, like the relationships between synaptic transmission and spike generation. And it's also about people and the colleagues and the patients that we serve. And our labs and our communities they're not merely collections of equipment and knowledge, but they're groups of people. And our scientific relationships are not merely transactional and utilitarian. They're rather, our scientific work is a labor of love. I mean, why else would people stay in these jobs until their 70s and 80s, and even until they die in some cases, as you know? Science is difficult enough, but without love, it becomes almost impossible. And, you know, like most people, I can be jaded and cynical at times, but what I'm realizing more and more is that we bring abundance into our personal and scientific lives through love. So I try to keep love in mind when doing science. And the second thing that I'm going to talk about is synaptic transmission, because good luck giving or receiving love without synaptic transmission. And thirdly, what is the point of synaptic transmission if not for spike generation? So today I'm going to give an overview of some of the things that we and others have done on these subjects. And I'm going to add in a little vestibular just to make it tastier. And I recycle some slides, but I never give the same talk twice. I'm definitely going to cover some work from other groups, and I won't be able to cover everything. And if I don't mention you or cite your favorite paper, I'm sorry, but it's not because I don't love you. So, hey, ARO, I got my Puerto Rico shirt on. All right, let's get started. So, first thing I'd like to talk about is the spontaneous activity in the auditory nerve, or the spontaneous rate, or SR. Just to make sure everybody's on the same page. This spontaneous activity in the auditory nerve is not endogenous to the nerve fibers themselves. And it's not spontaneous in the sense of the sometimes used term in synaptic transmission that it's not calcium independent exocytosis, but it does depend on calcium dependent exocytosis of glutamate, which is also referred to as synaptic release. And that release depends on the inner hair cell depolarization at rest by the MET current. And so the spontaneous rate depends upon synaptic transmission, and it's what's happening in the absence of an applied sound stimulus. And cochlear transmission is glutamatergic. And we know that most directly from experiments where brave people have patch clamped the let me turn on my pointer, where people have patch clamped the tiny postsynaptic boutons of the unmyelinated fibers that contact the ribbon synapse active zones. That's a cartoon that I drew, but it looks like this under good DIC optics from Grant et al. And this is an example of the excitatory postsynaptic currents that one can record from an individual recording. And if you put them all together into a binned histogram, you can see that there's a broad distribution of amplitudes up to about 800 picoamps, with an average of around 375, or a modal peak. And these are much bigger than the EPSCs from central nervous system synapses, which are about 50 picoamps. And so when you're recording these, they occur as a sequence of events that are randomly timed. And when you put on an antagonist of AMPA type glutamate receptors, it goes away pretty much completely. And so we know that these are mediated by AMPA type glutamate receptors. And so because of this, cochlear spikes depend upon quantal transmission. So this is our latest paper that just came out, which was a big collaboration. And we utilized the vGlut3 wild type and knockout mice, and we recorded in vivo from the auditory nerve. So you see this diagram on the right. This is sort of what the system is like in vivo. You have the auditory nerve fiber receiving an input from a single ribbon synapse, and here in the organ of Corti, the analog signal of sound is converted into a digital signal in the organ of Corti, and then that's propagated along the myelinated part of the peripheral axon by those nodes, and then into the central nervous system, and the recordings take place around here. And this is what a train of action potentials looks like in this example in the auditory nerve, in wild type in blue, and then in the VGLUT3 knockout, there's absolutely nothing. It's completely silent, and it's been known for over 10 years now that these mice are completely deaf, so this was not a surprise to see. But what we wanted to do was compare to what you see in the vestibular nerve on the right. So when we compared wild type and knockout, in that case, they look almost indistinguishable, which is interesting because this indicates that vestibular transmission relies predominantly upon non-quantal transmission, which I think is interesting. So we published this paper about the gross normality of the vestibular behaviors in these mice. And when we measured the timing and the rates of those spikes in the vestibular nerve, comparing the genotypes, we saw that the means were not statistically significantly different in terms of the mean spike rate or the interspike interval or the coefficient of variation of intervals. So auditory nerve needs it. Quantal glutamatergic transmission, vestibular nerve seems not to. And as I mentioned, this is not a surprise because the original reports from the mostly American group and the mostly European group showed that these mice are completely deaf. And as well, the system does not develop properly. So if you count the spiral ganglion neurons in Rosenthal's canal, in these SLC17A8 mice, which is the gene name for VGLUT3, they observed a reduction already at young ages. And again, this is in contrast to the vestibular system, where it's been seen that the vestibular afferent neurons develop normally in the absence of quantal glutamatergic input. And so we went ahead and showed that vestibular quantal transmission is solely glutamatergic, which had been previously postulated. So this is an experiment which I'm depicting in a similar study on the right, where the postsynaptic calyx is patched, but instead of a jet to move the hair bundle, the hair cell is depolarized by changing the potassium level in the bath from a relatively low level at 5.8 millimolar to a high level, or sorry, a normal level to a high level. This depolarizes the hair cell. The hair cell releases quanta of glutamate, and then you can observe the train of the excitatory postsynaptic potentials in wild type. In the knockout, there's no quantal transmission to evoke. So again, I think it's interesting to compare the auditory and the vestibular system. And it's not new, the idea of non-quantal transmission. One of the early papers looking at this by Heisting et al. In 2014 saw evidence of this. Okay, so now we're going to go back to the cochlea, and I'd like to talk a little bit about the anatomy of the spike generator. So this on the left is an adult guinea pig organ of Corti, and the neurons are labeled with sodium potassium ATPase. The hair cells are labeled with myosin 7A and also VGLUT3, which is robustly expressed in the inner hair cells. And where these unmyelinated neurons come together to go through little holes in the osseous spiral lamina called the habenula perforata, that is where they gain their myelin covering. And now I'm going to show you how that develops over postnatal development. And this is what a mouse looks like at P5 if you label for the presynaptic ribbons with CTVP2 and all the neurons with neurofilament and only the type 2 neurons with peripherin. In the relatively mature rat cochlea at P30, if you label for, I didn't put them on here, my fault, but the green are voltage-gated sodium channels, the red is Casper, and the blue is KV1.1. So these are the heminodes. I'm going to show you how those develop over postnatal development. And then you have an internode and then a node, and those are in the osteospiral lamina, and this is the ganglion here. So what we saw, I guess about 10 years ago now, with Kyung-hee Kim, is that the heminodes migrate to the habenula perforata around the onset of hearing in the rat cochlea. So I'm showing you the same three antibody labeling that I showed in the previous slide. NAV 1.6 is the voltage-gated sodium channel that brings sodium into the neurons. That labels the nodes and the heminodes. Casper is a paranodal marker, and KV 1.1 is a juxtaparanodal marker. And these histograms below are marking the positions of the NAV 1.6 clusters. So you can see at P9, they're not very organized yet. There's a little bit of a clustering closer to where the habenula perforata is. The organ of Corti would be here. But then at P10, P13, things are starting to get tight. And then right around the onset of hearing is where things really tighten up as these heminodes are migrating towards the habenula perforata. And then they continue to tighten up a bit more for about another 2 weeks, as you can hopefully appreciate here in these cumulative histograms. So we went ahead and labeled for a whole bunch of ion channels. All of the free antibodies we could get from SCBT at the time, because I had a guy in customer service working there that I knew, and he knew I was starting up my lab, and they wanted us to test out some antibodies. So, and this is a cartoon of the layout of this complement of ion channels. So anchorin G is found only at the heminode. NAV 1.1, I'm sorry, anchorin G is found only at the heminode and the nodes. NAV 1.1 was found only at the heminode. NAV 1.6 and KV 3.1b was found at the heminodes and the nodes, and Casper marks the paranodes. The part that's next to the paranode is called the juxtaparanode. That's KV 1.1, which is a low voltage activated potassium channel. And then KV 2.2 was found at the juxtaparanodes and also in the internode region, so the region between the nodes. And then along the entire neuron, we found KV 7.2 and 7.3 and sodium potassium ATPase. Okay, so now I'm going to switch up a little bit and talk about the relationship between synaptic transmission and spike generation. One of the things that has been fairly well understood for some time is that synapse position on the inner hair cell correlates with auditory nerve physiology. And I would say that it's fairly well, so the differences between the auditory nerve properties are quite well understood, but the relationship between synapse position is still being resolved. So on the right, you can see a figure from the canonical paper of single unit auditory nerve fiber recordings from the mouse, where we have raw data and a model laid on top of that of low spontaneous rate, high threshold fibers, and high spontaneous rate, low threshold fibers. So the low spontaneous rate, we're looking at the discharge rate as a function of tone burst interval. And the low spontaneous rate fibers can have a spontaneous rate that's near zero. And it takes a significant sound pressure level in order for them to increase their firing rate. They have a relatively broad dynamic range compared to the high spontaneous rate, low threshold neurons. So they have a significant spontaneous rate, which can be 100 spikes per second or higher. They begin to increase their firing rate at lower sound levels. They have a narrower dynamic range and they plateau earlier. So if one plots the relationship between the spontaneous rate and the relative threshold being the difference between the threshold on the audiogram and the threshold of those individual fibers, what one sees is an inverse correlation. So as the spontaneous rate increases, the threshold decreases. It's a fairly loose correlation, but it's consistently seen across species. And one thing that has been correlated with that is the position of innervation on the pillar versus the medullary side of the inner hair cell, where the medullary side ribbons tend to be larger and the pillar side ribbons tend to be smaller. Also, the pillar side neurons tend to be thicker and have a greater mitochondrial content. So one of the questions that I've been interested in for a while now is what are the synaptic mechanisms that contribute to this diversity of auditory nerve fiber properties? And I like to think about at least four potential sources of heterogeneity. Keeping in mind that in an individual hair cell, we've got 10 to 20 synapses, and they're all being driven by the same receptor potential, more or less. And the presynaptic and postsynaptic and efferent and spike generator properties are combining in ways that we don't fully understand to arrive at a variety of auditory nerve fiber properties. And that gives rise to this loose correlation between relative threshold and spontaneous discharge rate. And I think we can also think about those heterogeneities. We can think about those heterogeneities as giving rise to both the spontaneous and the driven rates. But I don't think we understand everything about that, but we do understand some things. And so on the next slide, I'm going to spend some time talking about some potential mechanisms of spontaneous rate heterogeneity. And so on the left here is kind of the way I like to think about it. And on the right are some recent findings that support those ideas. So what we have here on the left is three examples of inner hair cells innervated by two auditory nerve fibers each, one that's destined to be a high spontaneous rate and one that's destined to be a low spontaneous rate. And then we have three different scenarios for how we would get those differences in spontaneous rate. And so the first one up here is a different presynaptic release rate. So in this sort of thought exercise, the excitatory postsynaptic currents and perhaps also the excitatory postsynaptic potentials would be of the same amplitude distribution, and the neurons that are being driven have the same threshold for action potential firing. And in this case, because they have a different rate, I'm depicting a release event happening at this high spontaneous rate fiber, but no release happening at this low spontaneous rate fiber. And then on the right here, we have the high rate in condition A and the low rate in condition B. And because the neurons have the same threshold, but they're seeing different rates of EPSCs that are coming at the same amplitude distribution, the high spontaneous rate fiber is firing more than the low spontaneous rate fiber. And some of the recent evidence for this comes from Oskut and Moser in 2020, where they compared recordings between the pillar side and the medullary side. And what they found, what they're showing in this synopsis, is that when you depolarize the inner hair cell a little bit, you get some release from the pillar side synapses, but no release from the medullary side synapses. And it was previously shown by the same lab that this is most likely due to there being voltage-gated presynaptic calcium channels that have a lower threshold for activation on the pillar side than they do on the medullary side. Now, when you increase the voltage further, this opens up the voltage-gated calcium channels on both sides, and now the medullary side synapses are able to release as well. So this is an example of how a sensory cell diversifies its output by varying the calcium influx release coupling among active zones. And one consequence of this difference in coupling, or one way this is measured, is when one measures the relationship between release and the calcium influx. And they found that on the pillar side, this relationship is linear, most likely due to a closer association between the voltage-gated calcium channels and the vesicles compared to the medullary side, where it's superlinear, and because those calcium channels are further from the vesicles and it takes more calcium to be sufficient to bind to the calcium sensor. So in scenario 2, the EPSCs have a different amplitude distribution. So there would be a similar release rate in this thought exercise and still the same threshold for action potential generation, but the high spontaneous rate fiber would be postsynaptic to a synapse with a large amplitude of release, and the medullary side would be postsynaptic to smaller EPSCs. So these triangles are small here and large here, and even though they're coming at the same rate, they're only sufficient to evoke spikes most of the time for the high SR neuron. And this data comes from the work of Niwa et al. 2021. And what they're showing here is the release rate as a function of time when the inner hair cell is depolarized with high potassium, and they're again recording postsynaptically, as in the cartoon I showed earlier and as was happening in this example. So what they found is that on average, the medullary side synapses had a lower peak release rate and a more shallow slope compared to the pillar side synapses. And that's quantified here in H. Now, interestingly, if you look at P20, you don't see that because the pillar side synapses take longer to develop than the medullary sides, longer to mature than the medullary side synapses. So it really matters when you look. And these recordings are difficult to do at P20, and they're even more difficult to do at P30. So this is evidence for scenario 2. Now, scenario 3 would be different postsynaptic spike threshold. So in this thought exercise, the synapses have the same release rate and the same amplitude distribution, but now the high spontaneous rate neuron has a lower threshold that's depicted by this dotted line. So all these triangles are the same, but now the high SR neuron can fire more often because it's more excitable. And we have some unpublished data from 14 years ago now or so, where instead of recording EPSCs, I was recording spikes that I was triggering in current clamp with waveforms that were intended to mimic EPSCs. Actually, in this experiment, they were just square pulses. And the size of the square pulses is shown here on the right in the step size, going from 100 to 500 picoamps. In this sensitive neuron, what I'm calling the sensitive neuron, it spiked already for 25 picoamps with a relatively long latency, and then a briefer latency for 50 picoamps, even briefer for 100 picoamps. And then with increasing step current size, it kind of plateaued. It reached a maximum, it sort of reached a speed limit in terms of how fast that neuron could spike. So it would spike faster and it was more sensitive. And then if you compare that to this other neuron that I recorded from, it was relatively insensitive. So the smallest stimulus did not evoke a spike. It took 2 times as much current to evoke a much longer latency spike, and it never responded as fast as the more sensitive one. So these are 3, I think, plausible examples. And in my mind, these 3 things are most likely combining in different ways to get the full range of auditory nerve fiber response properties that we see in vivo. Now, these were from mouse, and rat. And I'm telling you that because now we're going to change, and I'm going to show you that species matters. I also told you that age matters, and also tonotopy matters. So on the left here, this is back from our rat study in 2016. And now we're looking at cryosections, radial sections. So here's the spiral ganglion, osteospiral lamina. Here's the heminode, the terminals of the neurons, and then an inner hair cell. And it looks pretty much the same, whether you're in the apical, the middle, or the basal cochlea. Now, this paper that came out quite recently, I think, is quite interesting. It shows that species and tonotopy matter. So, as we discussed earlier, there's this difference between the medullary side and the pillar side ribbon, and that's always been associated with differences in sensitivity. But what Kostranov and colleagues noticed is that in the guinea pig, the heminodes are actually in a different location. They tend to be in a different location depending upon what side of the inner hair cell those neurons are going to innervate. Where the medullary side neurons have heminodes that are further away than the pillar side neurons. So this would have the effect of changing the cable properties leading up to the spike triggering zone, which would tend to make these pillar side neurons more excitable. So this would go in the direction of having pillar side synapses being more excitable, having a higher spontaneous rate. But it's different from a presynaptic phenomenon related to the size of the ribbon. This is a postsynaptic phenomenon, which is like scenario 3 that we saw on the previous slide. And so when they looked at the relationship between the distance between the synapse and those heminodes as a function of the medullary pillar axis, you see a relationship like this where they become closer towards the pillar side. And this relationship looks to me like an even stronger relationship than the one of the ribbon volume across the medullary pillar axis. With the ribbon volume, what you see is that the largest ones are almost always on the medullary side, but the small ones can be anywhere. And this is, of course, consistent with the known fact that most of the neurons in the auditory nerve are high spontaneous rate. The low spontaneous rate ones are a smaller subclass. So this is consistent with the idea that you can have high spontaneous rate fibers anywhere, but it's really the low spontaneous rate fibers that tend to be on the medullary side, which we think are probably the ones that are more vulnerable to noise exposure. And then finally in D, this is looking at tonotopy. So it's the mean ribbon volume as a function of characteristic frequency, with medullary being in the dark color and pillar being in the light color. And so this difference becomes larger for higher frequencies. Maybe the fact that this relationship comes up again here on this side is maybe specific to the low frequencies at 8 kilohertz, and maybe that's why there's less of a difference here. Okay, and now we're going to go back to synaptic transmission. Doing all right on time. So this is some artwork by my friend Juan, and it shows our ribbon synapse. When the hair cell bundle is vibrated, we have a current that depolarizes the inner hair cell. This opens up voltage-gated calcium channels at the active zone. The calcium binds to the voltage sensor on the synaptic vesicle. It releases glutamate, goes across the synaptic cleft, binds to the AMPA receptor, evokes the EPSC. And because those EPSCs are happening very close to the location of spike generation, only about 10 or 15 microns away, and these things are happening at very high rates, there's a lot of ions flowing into the neuron. The pumps have to try to keep up and maintain homeostasis, but it's difficult for that to keep up sometimes, so water comes in, and this makes the neurons vulnerable to osmotic imbalance. And this is probably one of the ways that the neurons are damaged by noise. So let's talk about synaptopathy. So noise trauma causes synaptopathy, which is synapse loss or dysfunction. And if we look at how that looks in a mouse that's exposed to 100 decibels of octave band noise for 2 hours, you see that pre-noise, all of the pre and postsynaptic puncta are well paired. And 1 day later, there's a high degree of synaptic disintegration. And then if you look 2 weeks later, the debris has been cleared. Most of the synapses have been reformed, but they are fewer in number. So some of those synapses are permanently lost. And I wanted to test the idea that this is dependent upon glutamate before I just assumed it was the case. So we went back to the VGLUT3 knockout mouse, and we found that the knockout of VGLUT3, which takes away glutamate from the synaptic vesicles, was enough to preclude noise-induced synapse loss. And this is a pretty nice model because the vesicles still undergo exocytosis and release their contents because the voltage-gated calcium channels still locate to the active zones. But there's just no exocytosis of glutamate, and so there's no synapse loss, whereas in the wild types and the heterozygous mice, there is a lot of synapse loss followed by some repair or some recovery. And this is in black 6 mice, which is different from what was initially described in the CBA-CAJ mice, and that's probably a strain difference. You'll note that in the unexposed mice, there's already a reduced number of synapses, and that's because, as I said earlier, the system doesn't develop properly. There's a smaller number of spiral ganglion neurons and a smaller number of synapses. So we know that glutamate is involved in noise-induced synaptopathy, and one of the questions that came to my mind is, well, what about presynaptic calcium? In noise exposure, can presynaptic calcium be damaging to the hair cell? Because sound evokes presynaptic calcium influx, which evokes glutamate release, AMPA receptor activity, synaptic disintegration and loss. Also, this leads to some synaptic repair, which is associated with synaptic dysfunction, which has been studied by a few labs now in vivo. So we're going to label these synapses in the VGLUT3 knockout mice. This time we're going to label the voltage-gated calcium channels in addition to the ribbon and the AMPA receptors. And this is what that looks like. So we have the wild type on the left. It's actually a heterozygote, which looks the same as wild type. And the knockout on the right, unexposed on top, and 0 hours after exposure on the bottom. And here, what I'm plotting on the bottom is as a function of CTBP2 volume, either the CAV1.3 volume or the GluA3 volume in green or blue, respectively. And so in unexposed, in fact, in all of these cases, you always see a positive relationship between these two presynaptic markers, or between this postsynaptic marker and this presynaptic marker. But in the wild type, which is here on the left, the heterozygote here on the left, we see the emergence of a few new groups. So most of the synapses stay positively correlated like we see in the unexposed, although the GluA3, which is in blue, has been reduced in amplitude, so it's more overlapping the CAV1.3 now. But we also see things like this, which is shown in C1, which is where we have ribbons associated with presynaptic calcium channels, but no GluA3. So this is perhaps a rapid adaptation or removal of AMPA receptors from the synapse during the noise. And we see things like C2, where it's just calcium channels now associated with no ribbon and no AMPA receptors. When we do that in the knockout, we didn't see those groups appear. And so from this, I'm tentatively concluding that the presynaptic calcium influx appears to have little effect on the presynaptic structure, relatively little. Okay, for the last part of the talk, I want to talk specifically in more detail about the AMPA receptors. So AMPA receptors are tetramers comprised of GluA1, 2, 3, and 4, but in the cochlea, we don't have GluA1. It's a really interesting thing that we don't have GluA1. I'm not aware of any other synapse that's like that. Regardless of that, almost all AMPA receptors in all parts of the body have at least one copy of GluA2, and that's enough to make it impermeable to calcium. So by far, the vast majority of AMPA receptors are not permeable to calcium. But those that are lacking GluA2 are permeable to calcium, and they also have a greater sodium conductance. And the reason for that is because of the difference of a single amino acid residue, which is changed by a post transcriptional modification of RNA editing from a arginine to from a glutamine to an arginine. So that has a positive charge in the pore, and that's why most AMPA receptors are impermeable to calcium, because that RNA editing event is almost 100% efficient. But when that GluA2 subunit is not there, the positive charge is not there, and the channels are calcium permeable, as shown on the right. That lack of a positive charge that makes it calcium permeable also makes it so that positively charged polyamine compounds can get in the pore and block those calcium permeable channels. So that's the pharmacological that's the reason for the pharmacological selectivity of polyamine compounds onto calcium permeable AMPA receptors. So one thing I'd like to point out is that the postsynaptic density on auditory nerve fibers is very large compared to those that are seen in other parts of the nervous system. This is an AMPA receptor structure here, the pore-forming subunits that I was just showing you. It's about 15 by 15 nanometers, and in the postsynaptic density of auditory nerve fiber, you can fit over 2,000 channels if they're close packed. We figured out a way to label for GluA2, GluA3, and GluA4, and we find that all the synapses have all three of those. However, they have them in different ratios. So when you merge those three channels, you see that there's different shades of purple, and they also appear to be in partially non-overlapping domains. And this is just confocal microscopy, good confocal microscopy. And so when we label for the presynaptic ribbon, GluA2 and GluA4, what we always see is that there are these regions within synapses that appear to be lacking GluA2. So what we now know is that each synapse has GluA2 lacking calcium permeable AMPA receptors alongside the regular AMPA receptors. And we're going to take advantage of this pharmacology to prove that, because this is a synthetic polyamine compound which will block the calcium permeable AMPA receptor, but not it'll block the calcium permeable, but not the calcium impermeable receptor. And Juan showed this very nicely with postsynaptic recordings in the mouse, I believe this was. So he depolarizes the inner hair cell with high potassium. You see the EPSCs coming, and then he puts on IEM 1460. And after a minute or so, this reduces the amplitude of the EPSCs. You take it off, the blocker comes out of the pore, and the size of the EPSCs becomes larger again. So we thought, okay, cool. This is a neat way to block some, but not all of the AMPA receptors. And if synaptopathy is due to glutamate excitotoxicity, then we ought to be able to mitigate it by applying blockers of the calcium permeable AMPA receptors. And perhaps we could do this non-toxically, because we wouldn't be blocking all of the AMPA receptors, even if it's given systemically. So this was the first time, and this is working with Stephen Green in Iowa, that we showed that this was the case. And what you're looking at on the left is the response level of the ABR wave 1 amplitude as a function of tone input level. And when saline was injected, you see that before and after noise, there's a big difference in the wave 1 amplitude. But when this polyamine antagonist was on board, there was no reduction in wave 1 between before and after the noise. And this was correlated with a loss of synapses in the case of the saline, and a protection of synapses by the polyamine compound. And that's what's shown on the right. Okay, for the last part of the talk, we're going to go back to spike generation. So in this case, I'm going into current, well, right, on the top row, it's EPSCs, as I showed before. And this train of EPSCs, you can plot the EPSC amplitude as a function of the rise time. So you see we have some very small events, some very large events with different rise times. And I wanted to ask the question, what is the significance of having this wide range of excitatory postsynaptic currents? Like, what does it take to make a spike? So I simulated this range of spikes in current clamp by applying them as stimulations and evoking the action potentials, and then measuring the spike onset latency as a function of stimulus amplitude. The same way I showed you before with the sensitive and the insensitive neuron. And coming back to the idea that if it takes only 100 or 200 picoamps to make a spike, then why does the system appear to want to have such large EPSCs? This is very energetically costly. It makes the system vulnerable to excitotoxicity. But then you have to realize this is simply to evoke a single spike. A single spike is not enough to encode sound. For encoding sound, we need to evoke trains of spikes. So I did an experiment where I wanted to see if I could evoke spikes at 250 times per second. And for small stimulation, you only get one spike. For a medium stimulation, you get the first 5 spikes and then it skips a cycle and you need a couple and it skips. But if you do large ones, and now they're about 250 picoamps, which is just below the typical mean size of an EPSC, now we have well-timed spikes on every cycle. And if we quantify that in the form of a period histogram, you can see what I'm telling you. For the small stimulation, the timing is not very good. For 200, it's better. For 300, it's better. And so it seems like the benefit of large EPSCs to the temporal code is to improve the precision of spike timing. And the average EPSC is about 300 picoamps, so it seems appropriately sized to do this efficiently. And that brings us to the summary. So number one, love your science, or else nobody else will probably. Glutamatergic quantal transmission is essential in the cochlea, but not so much in the vestibule. The spike generator anatomy matures around hearing onset, like many other structures, but continues to do so for another 2 weeks. There are several mechanisms of auditory nerve fiber heterogeneity, but we don't understand all of them yet. Things like age and species and strain and tonotopy really matter. They're all interesting to study. And noise-induced synaptopathy is glutamate excitotoxicity that can be mitigated by blocking calcium-permeable AMPA receptors. And finally, the last point that I just made is that the excitatory currents are much larger than they need to be to evoke just a single spike. But at high rates, you need those large ones in order to prevent failures and to ensure spike time precision. And with that, I'd be happy to take questions. Or am I supposed to answer them online? Can't remember.
Well, we can do both. You can read the subject and you read the question in the Q&A, or if there are any raise hand, sorry, somebody who wants to ask question orally,
Sure.
that can also be done.
Yeah.
So,
Does anybody want to unmic and just ask a question? Or laugh?
okay, there's in the Q&A, there's a question about if you see any difference in the ratios of GLUA subunits in the base and different regions of the cochlea, like basal versus medial versus apex, and also where's any difference between pilar and modular.
So we have not yet had the chance to look tonotopically at the differences in those ratios. That's something that we should certainly do. I'm not sure that the mouse is the best species to do that in. I think I'd probably like to do that in the guinea pig. We have looked at medullary pillar differences in the mouse, mostly in the mid-cochlea, and we do not see a systematic medullary pillar difference in, for example, the GLUA4 to GLUA2 ratio. And if you plot that as a function of the synaptic ribbon, then you see a small trend. But at this point, I'm not convinced that it's consistent in one way or the other. But yeah, we're working on that. But if the relative abundance of calcium permeable AMPA receptors is a significant contributor to like the reason why medullary side synapses are more vulnerable, it's not an obvious one. So
Mhm.
it's something more nuanced than that.
Mhm. Good, thanks. There's a question by Wu, if you want to unmute yourself.
Yes, it's a great talk. I really enjoyed. I have a question about your vestibular recording. In your data, you showed, I think, a couple of fiber data. I'm curious which part of vestibular nerve you recorded from. Are they from like a superior branch, inferior branch? Are they from canal, otitis, regular, irregular? So that's part I'd like to probably hear more. Yeah. Thank you.
Yeah, so we don't know what part of the vestibular nerve that we were in, but we did see a range of regular and irregular firing behaviors. We just tried to go to the same spot in the wild type and the knockout.
Oh, do you get a chance to like rotate animal or translate animal or tilt animal?
No, we were just recording spontaneous activity in this study.
Yeah, thank you.
Any more questions? There's a question about mechanisms that could regulate synaptic loss during aging by Teshbih Kaur.
Hi, Tishbear.
Hi, Mark. Great talk, by the way.
Thank you.
I was just wondering, do you think if VGLUT3 or if glutamate is also a putative mechanism for the synaptic loss in the aging cochlea, or there could be other synaptic mechanisms?
Is excitotoxicity a mechanism for synapse loss in the aging cochlea?
I'm asking. I'm not sure. I think it's not. I don't know. What's your thoughts on that?
I don't know the answer to that question. Obviously, taking away glutamate accelerates synaptic loss. Having too much glutamate accelerates synaptic loss.
Hmm.
What we know from studies that I didn't have the time to talk about today is that if you take away one of the subunits, like GluA3, and this is published in two papers from the last few years with myself and Lanya Rubio's lab, what we have is a reduced overall expression of AMPA receptors and what looks like anatomically a shift towards more GluA2 lacking synapses. And this does cause synaptopathy, or what could be considered accelerated vulnerability to just ambient noise. So that could be considered aging, I guess, in a sense. And we also see a very similar thing if you take away the auxiliary subunit stargazing. And in both cases, it's specific to the females. So the synapse appears to be very sensitive to the stoichiometry of these AMPA receptor subunits. If you take away GluA3, not VGlut3, if you take away GluA3 pore-forming subunit, or if you take away stargazing, which is an auxiliary subunit, you get accelerated synapse loss, ABR threshold elevations, wave 1 reduction. You see the anatomical changes more molecularly in both the males and the females, but only the females lose their hearing.
That's interesting. Thank you. Thank you.
I have a question, if I may.
Hi, Stephen.
Hi, Mark. Great talk. I had a question actually from way back at the beginning. In the formation of the heminodes and the concentration of NAV 1.6 at the nodes, is that activity dependent? Does that also happen at the same schedule in, say, a VGlut3 knockout?
Okay, somebody studied that, didn't they? Raise your hand if you studied that. I mean, I have to go back and look in my folders. I might have studied that, but not published it. But did somebody publish that? Gabriel, are you here? Did Gabriel study that? Charlie, do you know? Help me. I got to phone 911 on this one. Thanks for the zinger, Stephen.
I don't know
Well-
that anyone's studied it, Mark.
Low-hanging fruit. I think I made some pictures and sent it to Gabriel and tried to get him to put it in a publication or something, but it never happened.
Hmm.
I'm pretty sure that was it. I think it is altered. I think it's delayed or somehow messed up. But yeah.
Okay, yeah, well, there's a paper for somebody out there. Okay, thanks.
Great. Well, thanks everybody for attending. It's been fun, and go ARO. See y'all in February.
Yeah, thanks, Mark. Very nice talk.